Yeah, so we are here today to tell you a story about imaging, but maybe a slightly different kind of imaging. And not just a story where the imaging and the imaging technique is at the focus, but also how the desire for pushing imaging actually also pushes us in other areas of engineering that we otherwise probably would have not gone into. And the particular part of imaging that Aleksandra and I are very much interested in is imaging of living things. And the challenge with imaging living things is that you want to image them in an environment where they're doing what they're supposed to do. Because if you cannot observe them in a dynamic fashion, well, if they're no longer moving, they're most likely dead. So if you want to understand the biology, if you want to understand the living aspects of it, it's very beneficial to observe the structures in their native state. Now that's super easy in areas of microscopic areas, and we have great microscopes go down to areas where we can really easily observe living things and biology in action. But there's a whole area that the science is exploring that goes at a smaller size scale where the tools that are available are not as forgiving in terms of imaging live samples. And that's around the area of bacteria, mitochondria. Obviously, we can see them, but it's difficult to really observe them for a long time at high resolution without disturbing them. And there's a technique that has at least the potential to work there, and that's the area of scanning probe microscopy. Now, scanning probe microscopy is a very broad technique, a broad category, and the idea of all of these techniques is that you have some kind of probe, something that measures something about the sample, and in order to get an image, you're moving that probe across the surface. And there's lots of different areas because this probe can be many, many different things. One of the most well-known and successful examples of scanning probe microscopy is the atomic force microscope. I don't want to spend too much time on it here. I wanted to show you some representative images and some examples why this technique is very, very good for nanoscale biology, because it has very good resolution. It has particularly the ability to get this resolution in an aqueous environment, in an environment where you can give nutrients, an environment where you can heat your samples to 37 degrees if you must, and feed it with CO2, whatever it is you need to do. So you can get images with this of the topography, but you can also get additional information because you're physically in contact with the sample. And the way it sort of works is that you have this sharp tip, which is the probe of the scanning probe microscope, and you're imaging with it and you're moving it across the surface to measure that property point by point. And so this actually is the first work in this area where Aleksandra and I had started to work together, because I came from the scanning probe AFM side, and she was doing great work in super resolution optical microscopy, and we were both trying to push it towards this live imaging at high resolutions, but from two different directions. And that's when we started to work together, and we got this correlated AFM super resolution microscope system going, where we could observe the living cells with both atomic force microscopy, and in this case it was STED, I believe, that was the palm, and the STED was the bacteria.
It was a long time ago.
Oh yeah, it's been so long ago. Okay, fine. So in case you put this on YouTube, cut that section out. So we thought this was really, really nice and really interesting, but it was really not satisfactory because it was still taking too long, and the goal that we had of observing something that was relevant without killing the cells was just not achieved. And one of the problems that we're having is that because we were trying to image point by point with our probe, it just naturally takes a very long time because you have to go every point individually. And that's why if any of you has done any AFM or any other kind of scanning probe microscopy, you get a line every second or slower, and it just takes a long time to get the full image. And during that time, your sample has changed. So the beginning of your image, it shows a different state than the end of your image. Now, there's been a lot of research in the last almost 15 years on improving the speed. It's called high-speed atomic force microscopy. I don't want to spend too much time on all this great science that has been done, but I just wanted to point out that this is a field that has been around actually for quite a long time. It's been for 15 years. But it has not really made it into the broad applications, the broad labs, because it requires quite a lot of custom instrumentation. It requires quite a lot of technology. And here I want to take a small detour towards something that's very dear to my heart, and that is to show that in order to get images of techniques that are not straight out of the catalog, you have to develop a lot of instrumentation. And if we are developing this instrumentation and then we use it to get a couple of nice images, that's great for a paper, but we have not really made an impact in the society or in labs. So that's why we've started in my lab to actively share all of the technology that we're developing. We're actually building the instruments to a point where other people can copy them. And actually people can come to our lab to learn how to build them. Here's an example of one of our high-speed AFM incarnations. Just for reference, it's about the size of an espresso machine. And people come and want to build it, take it home with them to do their own research. So this is an important part that I wanted to point towards, is that it's not enough to just get the pretty images. Our students work very hard to get those images. And if the outcome is just the few data points that we create, we are not really making use of the full technology. So now we're trying to have this program, and it's also supported by EPFL, to share not just our images and our image data, but our full imaging technology. So now getting back to the science, our high-speed AFM is slightly different than others. And one of the enabling features that we have is that we are trying to increase the speed of the imaging by increasing the speed at which we collect the data at every data point. And the clever thing that some of the students did was that they said, well, it's very difficult to physically move the probe to each data point, move it away, go back in and measure. That takes a lot of time if you do it physically with motors or piezo's or stuff like that. But what's very fast is to operate diodes, laser diodes, light sources. And so what they did is to add an additional laser source into our AFM that is focused on this cantilever. So this was this probe that you saw scanning earlier. The first laser spot at the front is very standard. We need that one to actually measure the tip-sample interaction. But then they've added another laser spot at the back that is there to actuate it. Because it's a very, very small MEMS device and consists of silicon nitride and a metal layer on top. If you heat it with the laser spot down here, you get what's called a bimorph motion. And we can pulse such a laser diode very, very fast. And this cantilever is very, very small. So this sampling of the surface can happen very, very quickly. And that way we were able to increase the data acquisition speed of our microscopes by 2 orders of magnitude. And that was a big deal for us. And in order to do that, we had to do some typical instrumentation, but we also had to innovate in the area of microfabrication. So in order to get this up and down motion in an effective manner, students were actually working here at EPFL in the CMI to develop special cantilevers that are optimized what is biomorph effect. So this is a whole area that was development in the lab that was kind of like triggered by the need to get nicer images. But it's not an imaging area, it's a microfabrication challenge. So enough about the technology. Here's just something that we can do with it. So after we built these microscopes, it's not enough to just look at calibration samples. We want to have something that actually people are interested in. This is a nice thing here. Both Aleksandra and I, we are in the Institute of Bioengineering, which spans the School of Engineering and the School of Life Science. So we have access to a lot of interesting colleagues, a lot of interesting samples. And Pierre Ganzi is one of these colleagues who has this sample that he studies, which are centrioles. And centrioles are organelles inside all eukaryotic cells that are very, very well designed for their structure, for their symmetry. And if you want to know more about it, go to Pierre's talks. He knows all about this. But one of the core things is that at the center of the centriole, there is a ring of proteins that nucleate the whole growth of those. And what Pierre was looking at was, how does this actually happen? What's the heart of this creation of the centriole? And they give nice electron microscopy, and you can use them AFM, where you see how these proteins, they're like dimers with a little head group, how they form these rings. But you only see the end results. You don't see why they do it or in which mechanism they do it. And so together with Pierre, one of my students was looking at this, and he used this technique to really resolve the imaging, resolve the assembly process on a single molecule level. So here you see these little wigglies that look like, well, they look a little bit like spermies. They come in and they self-assemble into this ring structure. And from the fact that now we actually see it happen, we found a whole set of new assembly mechanisms. So this imaging development has now triggered a whole area in Pierre's lab where they're trying to find models and different computational methods to explain why they assemble in this way. Another collaboration that we have is with Maciej Weestings from the Materials Science Department, where we're looking also at molecular self-assembly on, this case, with DNA tiles. And her area of expertise is how do you create new materials, programmable materials, based on the well-controlled and well-known structure of DNA. And what we did, we created this little DNA tripod, threw them all together, and they're supposed to form these hexagons. And now we're trying to understand what determines this reaction, what determines this growth. And we can now observe this in real time. And we can actually extract from the imaging sequence, we can extract now reaction kinetics at the single molecule level. So this is an example where the imaging part was actually, just the beginning. And the fact that you can have these images now triggered a whole new direction of the research that we were doing. For example, now there's a lot of automated image processing that came out of this, because now we have all of this data, and we want to see where are the hexagons, where are the defects, and things like that. However, all of the things that I just showed you were not living things. And I started this whole thing because I wanted to look at living things. I was just looking at molecules. The problem, if you apply the same advances and the same technologies to smallest living objects, cells, is that the atomic force microscope, as the name already says, requires a force interaction between the object that you're looking at and your probe, the cantilever. And if you do that with a eukaryotic cell, for example, well, you're not actually probing the surface. You're deforming the surface until you probe something inside. And that's been a fundamental problem in applying atomic force microscopy and scanning probe microscopy in general in cell biology and the life sciences. Fortunately, much smarter people a long, long time ago realized that you could do other things. And this was a paper that came out only a few years after the invention of the atomic force microscope. So it's actually almost as old. And Paul Hansma and Barney Drake developed what they called the scanning ion conductance microscope. And the idea behind the scanning ion conductance microscope is just to replace the force probe with another kind of probe that you can use to detect the surface, but that does not rely on a force interaction. And the probe that they were using was a glass pipette. And so here, 30 years later, we use the same thing, just better graphics. We have glass pipettes with very, very small holes. Inside the glass pipette, there is an electrode. Our sample sits in an aqueous solution with ions, and we have a counter electrode. Now, if we apply a voltage between this inner electrode and this outer electrode, then ions will want to flow. And they want to flow from here out through this aperture all the way over here, get to this electrode to create the circuit. So if we are applying a voltage and we measure this current, we will see that the current is determined by this whole loop. Now, the current is determined by whatever resistance we have here coming from the electrode out through the path. Then the resistance as it goes, the ions come from here to here. But it also depends on the resistance that the ions physically feel as they're coming out of the tip past the sample. So you can think of it like this, and normally I bring a tube, and I forgot I have to say, I'm sorry, I have to just do it like this. Let's assume this is my pipette. It actually has a little hole at the end. And let's say I'm here in free space, and ions flow from this electrode to this electrode, and it just flow through here. And that's perfectly fine. They can do that. But now I bring this pipette here to the surface. And the closer that I bring it to the surface, the smaller is the gap for the ions to go through. So the harder it gets for them to actually move from the inner electrode to the outer electrode. So the closer that I get with my pipette to the surface, let's say this is the distance from the pipette to the surface, the less current I get. Obviously, when I'm really on the surface, I get zero current. Up here, I get the basic current. And in between, it drops. And because I know how it drops, I can now detect the surface even before touching it. Huh? So we have a nanoscale proximity sensor that detects the surface without ever actually having to have a force interaction. And that's very, very nice, because now you can image things that are super soft. With normal AFM, there are cells that you can look at, but they're more the stiff cells. The soft cells, neurons, stem cells, they just really don't like to be physically touched. And that's the area where we developed the scanning ion conductance microscope for. Here's an example of excuse me, some neuron cells. And just to show you how we do this. Again, we have this pipette. As you can see, we are steadily progressing in the quality of our animations. Right? Current comes down, and we keep going down until we reach a certain threshold. We haven't touched yet, and then we go back. And then we do it again, and we do it again. And then we measure, we keep track of how far we had to get down to get to that threshold. And then the envelope of these thresholds then corresponds to the outside of our cell without us ever having to touch it. So this is really great. It's super, but it's hella slow. Because not only do I have to detect it at every point, I have to be very gentle coming down so that I don't overshoot. And so this took us away from our idea, yes, now we can image living cells that are very fragile, but we could not do it fast enough. And maybe I should just glance over this and looking at the time. But the problem is very, very similar to what we had been facing with AFM, is that we need to do this quickly. We need to actuate quickly without overshooting and interacting too harshly. So we could apply a lot of the things that we had learned from high-speed AFM also to this. Unfortunately, here we could not do this trick with making custom cantilevers and lasers and things like we had before. Here, we actually had to come up with a few new things. And just to cut a long story short, there was a lot of development in many, many areas in terms of mechanics. So this is nanopositioning scanners. Up here, there was a lot of effort in creating an environment where the cells can be kept alive. There was a lot of development in electronics. We even had to go to some custom IC manufacturing to get up the performance, a lot of programming. But putting all of these things together now gave us the opportunity to image in a much more reasonable time frame. The amount of time it takes us for imaging depends a little bit also on what you image. If you want to image a very large area because you have to go to the different areas, it takes you longer. But now, even for very large areas, it takes us a few minutes to image. If we go to smaller and smaller areas, we can get an image every couple of seconds. And I see Samuel smiling because this slide is horribly outdated. Now he gets several images per second. Yeah? And what we can do with that now is to really start to look at the living cells in the native environment without bothering them too much, without distorting them, and observing them for a long period of time. And I'm not going to go at all into the science here. I just want you to look at these images, because for me, they were just, maybe I'm just biased here, but they were showing cells in a way that I had never seen them before. I've seen images like this from scanning electron microscopes, see 3D structure of the cells, but they just were dead. And I kind of like to look at these cells or this imaging mode now in a way that we can like spy with a little nanoscale video camera on the cells without them complaining about it, and really see how they develop, how they grow. And now we have the ability to treat them with different kinds of drugs. We can do other kinds of distortions. Really, we can investigate a lot of the behavior of the cells, not just the structure, but the behavior and the responses to stimuli. I'm pausing a little bit for you guys just to take it all in. So what you're seeing now here is the surface. You see the topography. But any biologist that we talk to, they say, oh, yeah, there's very nice features. What are these? What's underneath? What's inside? Because they don't just want to know what these things look like, but they also want to know why do they look that way. And unfortunately, this is one of the problems that scanning probe microscopy has. And also scanning ion conductance microscopy has that, because we are sensitive to the sample surface. And in order to get around that, I'm going to hand over the microphone now to Aleksandra. It shows you where we can, again, pull our resources and do way more than either of us could have done by ourselves. You want to take this? Or you want to stand here?
Yeah, I will stand here. So leveraging on the hard work from Samuel and the collaborators from LBNI, and also having a great couple of students, so Vytautas and Samuel. So they teamed up to combine these two techniques and indeed peer inside the cells. So similarly as we did with the atomic force microscopy and correlating it with the photoactivatable light microscopy, this time also taking advantage that there has been a big progress in a speed of SICM, we also had to correlate our images with the modes of imaging in super resolution microscopy that temporally also matches the timescales of the scanning ion conductance microscope. So we sacrificed for that a little bit on the resolution side and decided to work with a modality in a super resolution microscopy that also relies on a blinking fluorophores, which is called super resolution optical fluctuation imaging. And instead of localizing, I guess many people here are familiar with localization type of microscopes, we correlate the blinking between the neighboring pixels, and using special mathematical tricks, we can push the resolution of the microscopy further. So looking at this cross correlations. So we exploit these higher order statistics of a time series, so meaning that we need to take some temporal progression of the images, and basically improve the resolution by the square root. So the order of the SOPHY is defined by the by this cross cumulant, and the resolution is defined is then improved by the square root of the order of the cumulant that we take when we do this type of cross correlating work. So the higher order of the SOPHY requires the more images. So again, here, taking the same movie, we can kind of play with the temporal resolution in expense of the axial resolution. So I hope I get this across. And one very nice thing about the SOPHY is that it also tolerates relatively high density of the fluorophore, so we can have also imaging a very densely labeled species. So here again, in collaboration with Georg's lab, we combined the two microscopes on the same platform. So on the typical optical microscope, we could place a nice compact scanning ion conductance microscope, and in the same time or sequentially acquire these two types of imaging modalities. So we also profited a lot from the development of the dyes from our collaborators, who were able to get us a very nice dyes that target the actin. And then just looking at the images in a SOPHY modality, we see this improvement in resolution as we increase SOPHY order. So independently as well, then we could do on the same sample imaging in a scanning ion conductance modality, and as well in SOPHY modality, and cross-correlate the same images and get this nice topography together with information about structures that one can image inside the cells. And here we looked at the microtubules, at the actin, and nice topographical information that we achieved from the scanning ion conductance microscopy. So one thing that we also innovated in the lab is acquisition of the SOPHY images simultaneously in 8 planes. And for that, we take this custom-made prism that was also made by our collaborator, Theo Lasser. And in the same time, we can acquire movies in 8 sectioning planes across the cell. So that in that way, again, using the scanning ion conductance microscopy, we can combine these two modalities, and also not get the projection from 2D projection, but also 3D volumetric imaging in a SOPHY in combination with scanning ion conductance topographical information that arises. So one can see this movies that then one can do with combining this imaging modalities. And of course, we can do all this live on the same platform. So another thing that was very long on my research agenda. So it actually started in 2003. So at that time, I was a finishing PhD student, and I worked actually on a cryo atomic force microscopy, motivated by the nice work of Dubochet. So my goal in a PhD was to develop a cryo atomic force microscope. But then again, looking everything that was dead. 4 years passed, and my PhD was not such a great success as a cryo-electron microscopy success, and I felt really as a failure. And as my postdoctoral work, I wanted to work in more physiological conditions. And actually, I proposed a method where one would combine nanopores and force spectroscopy in order to control translocation speeds through this nanopores. Nanopores were arising at that time as a method for sizing molecules, but also people have dreamt in 2003 to sequence DNA. Now we know this is a reality, so we can sequence DNA with the nanopores, but it was a really long road. So coming to this historical perspective, so 2003, idea of combining the tweezers to control the speed through the nanopores, and having neighbors who work on scanning ion conductance microscopy using the pipettes that are in fact nanopores, really gave me idea to think about why are we maybe wasting the time combining everything with the optical tweezers. Maybe we should really do straight deposition of the molecules on the surfaces and exploit this nice technique and the way that we have progressed technologically in terms of not only instrumentation, but also probe development. So going again a little bit in the history, even further in the history, because I think it's good to remind people about historical developments. So the pipettes have been used for studying ion channels, and they have given people a Nobel Prize. So here are pictures of Bert Sachman and Erwin Nair. So they looked at the functioning of the ion channels using precisely this quartz or borosilicate pipettes. By patching the membranes, they could measure the activity of ion channels and tell us really open up a new area of research that has, I think, revolutionized neuroscience. So another technology that I also briefly mentioned are nanopores. So they are, again, in this nice animation, they are method that allow us to read the information on a DNA. So similarly as the nanopipettes, we can also think of the nanochannels in biological pores or solid-state nanopores as a small holes through which ions are passing, but also sometimes single molecule can pass through. So as ions are modulating as well this base current, that Georg also nicely showed in his signal generation, the current can be also modulated once the molecule also passes through the pore. And in the heart of the nanopore sequencing actually lies a biological pore, which is in fact coupled with biological motor that regulates the speed at which this molecule passes. In the early beginning of the research with the nanopores, people have been hopeful that they will have electronics that will be sufficiently fast to measure the signals for these free translocations. But unfortunately, this didn't happen until there was a nice idea from a colleague, Jens Gudlach, to couple the biological motor that then pulls on the DNA and regulates this speed and allows electronics to really read this minuscule changes in the current that are linked to the changes in the sequences. So my goal in 2003 was actually to have engineered or go away from this biological motor, but have some way of controlling this translocation speed. And as I said, we tried many, many different things, from optical tweezers and magnetic tweezers in other labs, and atomic force microscope in other labs, but nothing was working so well. So coming to the successes of the nanopore sequencing, so it is really successful technology, but it still has a quite high error rate compared to Illumina or optical methods. And but it has many advantages. So it can sequence very long pieces of the DNA. It can reread the molecules, but not at the stage that I will show you. So it can be they can be reread twice or a few times. And one most important part is that it relies on biological motor. So this biological motor is in no way under our control. So this biological motor, as anything in biology, is a stochastic motor that behaves at its own will, and we cannot choose which molecule to pick. We cannot choose at which rate to work, and we cannot choose how often we want to scan the same molecule. And on top of that, if we want to rescan some small area of the molecule that we would like to study in a greater detail. So as I've stressed, so the motor is really stochastic and not under in any ways under our control. So just to mention, so there is a big research in biological pores, and they are up and coming, and people are very excited about them because again, they can one very good advantage over what we can make as engineers is that when they are sufficiently small, so sizes of the MSPA, for example, or irolysine, they fold almost always identically. But as we move to the larger sizes and more complex structures, this folding is not always as precise. But concentrating on the pores that are used in sequencers, they are pretty much, I would say, tour de force of engineering that nature can do at a really nanoscale. So, so they are provided actually for free, so we don't need to engineer them. There is also great effort from the community that looks at the protein design, and I think that's the area that one needs to look at and also keep an eye on it, and that's the way that one can now also engineer likely pore of its pore that has a property that one would like to have. But it's still really in its infancy. And at the moment, what the nanopore community does is takes these biological pores and mutates a few amino acids in order to change surface charges and to provide a better sensitivity. So coming from the engineering point of view and from the fact that where we used these capillaries that you have seen as well as the imaging probes, so we have developed a process in 2013 by which we can shrink these pooled capillaries that you have seen that have been used also by the researchers in the patch clamping. So they were able to pull the capillaries to specific sizes, but achieving the sub 10 nanometer diameter of the capillaries just by pulling is still very, very difficult. And it also comes with some drawbacks, which give the high aspect, for example, high aspect ratio and very large, very large pulling areas where when we pull. So in terms of geometry, it is better to pre-pull these capillaries using the pullers to the bit larger sizes, and then to shrink them in a scanning electron microscope as we do. And as for the scanning ion conductance microscope, for our technology where we look at the molecules, the smaller capillary means a higher signal to noise ratio, because we are matching the size of the analyte that passes, and as it blocks the pore and modulates the current, if it's closer to the size of the capillary, then we have a higher signal to noise ratio. So these nano capillaries have been also used to look at the signals from the topologically structured 1D DNA origamis, such as these dumbbells. But as you can see, sometimes it is very difficult to distinguish multiple wells that one has, and the signal to noise ratio is not that great. So clearly, combining the ability of scanning ion conductance microscopy to really pick a single molecule from the surface and then controllably scan on it would be beneficial in order in many ways. In the normal translocations, as I mentioned, we have a low signal to noise ratio, and we cannot. It's very difficult and very challenging to rescan the same molecule. As already pointed in a slide that I showed before, so the method relies on the molecules that are chemically attached to the surface using a streptavidin biotin linker or PEG linkers, and with the scanning ion conductance pipette, we approach to the surface, but not in a dangerous regime where we can crash a pipette. But sufficiently close for a DNA molecule to sense the electric field that is placed across the pipette and enter through the same entrance as the ions are typically flowing in scanning ion conductance modality. So here we also have all the time ions, millions of ions also flowing, and now occasionally when we want and where we want, we can also catch a single molecule. Just to showcase, so the method is also compatible with physiological conditions. And as a first test sample, because we could position our capillaries are also tunable to the sizes, so we can make capillaries that can accommodate also DNA protein complexes that are relatively larger. So using the nice technology of dCas9 and the guided RNA, we can position a protein to bind at any location that we programmed using the guide RNAs. And then using the scanning ion conductance microscope operating in a spectroscopy mode, catch the molecule, and then detect such a Cas9. And again, as we have everything built on optical microscope, we can also profit from correlated fluorescence imaging. But coming to the question of how much we can push the resolution and increase the resolution. So for that, I contacted a collaborator who actually introduced this dumbbells as a ruler for benchmarking the nanocapillaries and solid-state nanopores in terms of the signal-to-noise ratio, and asked him that we perform an experiment in which we would benchmark our system. Looking now on the free translocation state-of-the-art that our collaborator has here operating in a 4 molar lithium chloride, we see the signatures of 6 dumbbells that because they are translocating freely, there is a temporal variation in which we see that they occur. And on top of that, although they are topologically should be the same structures, we see that they are not modulating the current in the same way. Unlike that, in the controlled translocations, we see a clear improvement in a signal-to-noise ratio. And on top of that, the distance. So since we are on control, we are in control of the position. There is a really nice and the same distribution between the sizes, because indeed in the folding design, these dumbbells are placed equidistantly. So unlike in this temporal information that we see here. So what we could also vary, we could vary here a voltage. So also unlike biological pores in solid-state pores and the capillaries, we can go also relatively high in the voltages, but also in the taking advantage of a controllable speed. We could also test different speeds and see at which speeds we obtain the highest signal-to-noise ratio. So the slower we go, of course, the better signal-to-noise ratio. And so this is plotted on these slides. So just for comparison, we can look also on the distribution of the controlled translocation So these are these distances versus the free translocation, and we see really a clear improvement. So we could then, I'm thinking about Samuel. So Samuel can go from one molecule to another. So here, looking at the 100 different single molecules and controllably scan on them. But he can also choose to pick a single molecule and rescan this molecule hundreds of times. Similarly as in localization microscopy, here we can improve our signal even further by summing the signals. So going even further in determining the resolution of a system, we designed kind of another molecular ruler based on a DNA, where we introduce this variable sized gap from 80 down to 12 nucleotides. And again, looking with the capillary and the method that we have, we could scan these gaps in controllable way, doing here more than 1,000 readings of the same molecule, and in this case, replotting this area where is the location of the single gap. So in ideal case, for ideal capillary of very small sizes, one would expect here a plateau, but we have, let's say, also like this little bit convolution from the capillary that we see. So clearly, one would like to have to work with the smaller capillaries and better defined pores. But for us, was really amazing to see that the drop in a signal where we have a single-stranded DNA region is actually really half of the conductance drop that we have for the 2 strands. So really, in my view, very amazing result. So then just to also show that method can be multiplexed on the same sample, we spotted different sizes of the gaps, and we could see as well, we could resolve these gaps also with our capillaries. Clearly also seeing that our resolution and will definitely improve if we choose to work with the smaller capillaries. Or, as I will tell you later, implement some other tricks. So going even further, so using these gaps, we can hybridize the DNA either in a test tube or later in a situ just by choosing the complementary sequence. And now looking at the possibility to observe this single base gap in a DNA, and just by operation that we can rescan the same molecule many times, and working with the small capillaries and with very talented students, these results can be obtained. So what we would like to do further as the resolution of any microscope, but this one as well depends on the probes. And to get smaller capillaries, I believe we exploited some of the ways, but using scanning electron microscope, this is where we stop because of the charging, so we cannot go further down. But embedding the biological pore or another type of material that we work in the lab as 2D material with a very small probe would likely even further push the resolution. So in our approach, since we are tunable on the size, we can really examine different types of combination of the single molecules, so the DNA plus proteins, or single-stranded DNA, or double-stranded DNA, RNA, anything that one can imagine. So showing as well and leveraging on capability of the scanning, we can also spatially address molecules. So there is a big advancement in the field of microarrays where molecules can be spotted, and in molecular diagnostics, this is a very well-known tool. So we can envisage that this technology is compatible with microarrays as well. So what next as well, and what one could also dream of, is to look at the also likely on the post-translational modification of the proteins, because the method is inherent, or peptides at the beginning. The method is inherently working in physiological conditions, and we can assess the same molecule after modifications. Of course, a method is compatible with many other optical techniques, and I think this is just the start of many nice experiments that we can do. So with this, I will give back conclusion for the part of the scanning ion conductance microscopy part, and I already give an outlook for the 6.
I would say, I mean, the conclusion, we're just presenting data, we're presenting images. I would say the one conclusion I would like to make in all of what you've seen today is that the imaging is both the goal as well as the enabling technology. But in order to get the imaging, there's a lot of other technology that needs to be done. So I, for myself, I do not consider myself a microscopist. I consider myself an instrumentalist. I like to bridge technologies or advanced technologies to a point where they can improve imaging. And the reason why I'm happy to be here and part of this imaging center is that that's only one side of imaging, is the image acquisition. The other side is the sample preparation, which is happening with lots of people that actually I consider the real microscopists. And then the next part is the image analysis and the image reconstruction, where there's also a lot of expertise here at EPFL. So that's why I think it's very exciting. And thank you so much for all the work that you're doing as part of the Imaging Center. I think it's a great initiative. And yeah, we hope to continue to contribute. And now, if you have any questions, I'm sure Aleksandra will also be more than happy to try to answer them. And of course, none of this stuff would be possible. I mean, we always say we can do this stuff, right? That's the royal we, because neither Aleksandra nor I are spending time in the lab operating the instruments. Here's in my lab, I would say Samuel is the key person behind all of this work. Barney Drake was here for a while. He's actually the inventor of scanning ion conductance microscopy. He really helped design and build the instrument. And now we have a new generation of students join the effort. And also in Aleksandra's lab, I don't know, you might want to.
Yeah, I already singled out Vytautas for the work on both projects, and now continuing Helena. But I also want to mention people who worked on the development of the nanocapillaries and also combination with optical tweezers. Some things, these things maybe were not so successful, but they paved the way for what we have now.